Thank you for the opportunity to present to you today an overview of ethical considerations in pediatric clinical trials. Over the past 25 years, we have come to understand that we must protect children through research rather than from research. This need for pediatric research reinforces our responsibility to assure that children are only enrolled in research that is both ethically sound and scientifically necessary. I'd like to call your attention to two recent FDA draft guidance, one of which is exclusively about the ethical considerations for pediatric clinical trials, and the other of which contains a section on ethical considerations in clinical pharmacology studies in pediatrics. This presentation is based on previous work of myself and is consistent with these draft guidance. And although the focus is on the U.S. ethical framework as reflected in the FDA regulations, this approach is generally harmonized with international pediatric guidelines. The starting point of an ethical evaluation of a pediatric protocol is that children should not be enrolled in a clinical trial unless their participation is necessary to answer an important scientific and/or public health question about the health and welfare of children. This is referred to as the ethical principle of scientific necessity and is grounded in the principle of social justice and derives from the requirement for equitable selection. The practical application of this ethical principle is found in pediatric extrapolation, and I'll call your attention to a recent ICH E11A draft guidance that has been released on this topic. In addition to the ethical principle of scientific necessity, there are three other principles that are reflected in the regulations. The first is absent a prospect of direct therapeutic benefit, the research risks to which a child is exposed must be low. Second, children should not be placed at a disadvantage by being enrolled in a clinical trial, either through exposure to excessive risks or by failing to get necessary health care. And finally, vulnerable populations unable to consent for themselves should have a suitable proxy to consent for them. The two ethical principles about risk minimization are reflected in the additional safeguards for children found in the FDA regulations of 21 CFR 50 subpart D. There are two components of this regulation. First, absent the potential for direct benefit to the enrolled child, the research risk must be low. In the FDA regulations, this is divided into two categories: minimal risk or a minor increase over minimal risk. Otherwise, if the risks are greater than low, the child must have the possibility of a direct benefit that in fact justifies the risk, and this balance of risk and benefit should be at least as favorable as any available alternatives. Note that we cannot expose a child to greater than low risk for a benefit to society, as suggested in ICH E6R2, but this was modified by R1, noting that in fact there should be a direct clinical benefit to the child, or the risks must be restricted to low risk, as found in ICH E6. There are two key concepts that rest behind this ethical framework. First is the prospect of direct clinical benefit, which basically means that the experimental intervention or procedure should improve the health or well-being of the individual child. So the assessment of direct clinical or therapeutic benefit is an essential aspect of the ethical acceptability of a pediatric research protocol. Now, in addition, a protocol can contain multiple interventions or procedures, some of which offer a prospect of direct clinical benefit and others do not. These different components of a protocol must be evaluated separately to assess whether the risks of those components do not offer direct clinical benefit are appropriately evaluated as meeting the criteria of low risk. Why is this important? failure to carefully distinguish different components of a clinical investigation may result in the risks of an intervention or procedure that does not hold out the prospect of direct benefit, exceeding the allowable ceiling of low risk, or as the language is found in the FDA regulations, a minor increase over minimal risk, absent referral for federal review, which is a pathway that's available in the United States. Examples of the types of procedures that should be analyzed to assess the risk level include research-only biopsies, imaging studies such as MRIs that involve procedural sedation, and the risks of placebos, especially invasive ones that might include multiple injections or the use of a percutaneously placed central catheter. Now, when we link science and ethics, the ethical challenge is to establish sufficient evidence using preclinical animal models and/or adult human clinical trials to conclude that, in fact, we either have sufficient evidence for a potential prospect of direct benefit to justify the higher risk pathway, meaning risks that present greater than low risk, or that, in fact, we have sufficient safety data to be able to say that absent a prospect of direct benefit, the risks are appropriately low. These two pathways I like to refer to as the low risk pathway if there are no benefits, or the higher risk pathway if in fact we've been able to establish that there is a prospect of direct clinical benefit. Now, low risk in FDA regulations, as I mentioned earlier, the language is minimal risk or a minor increase over minimal risk. Minimal risk is defined as the risks that are ordinarily encountered in daily life or during the performance of routine physical or psychological examinations or tests. Notably, when the regulations were first proposed back in the 1970s, the definition of minimal risk was meant to be indexed to the life of a healthy child. This is not in the regulations, but I was pleased to see that the newly released FDA draft guidance does say that minimal risk should be interpreted as though those risks encountered in the daily life of normal, average, healthy children living in safe environments. Now, administration of experimental drug or biological product is generally not considered minimal risk. As a possibility, we may have sufficient data to support that such administration is no more than a minor increase over minimal risk, or could be considered a low risk. But this is limited, if that's the case, this research is limited to children with the disorder or condition, absent again review by a federal panel. But in the FDA guidance, it also notes, as was stated by the National Commission in the 1970s in the United States, that a minor increase over minimum risk should only mean a slight increase over minimal risk that poses no significant threat to the child. I was also pleased to see in the clinical pharmacology guidance that was also released in September 2022 that there was an explicit acknowledgement that the FDA has the authority to place a pediatric protocol on clinical hold if in fact it fails to comply with the ethics regulations. Clinical holds can be placed if subjects would be exposed to an unreasonable and significant risk of illness or injury. And in fact, the argument is that the ethics guidelines as found in the regulations of 21 CFR 50 subpart D frames what would be considered an unreasonable and/or significant risk if in fact a protocol is out of compliance with these ethics guidelines. When I was at FDA, there were clinical holds that were being exercised, but there had not been a guidance as yet that explicitly stated FDA's justification for doing so. Finally, some words on parental permission and child assent. Parental permission is framed simply as the same approach as informed consent, but we use the concept of permission since one can only consent for oneself. Often we find the language of informed consent is used to encompass the idea of parental permission. Now, child assent means the child must give an affirmative agreement. In other words, simply being silent does not mean the child has assented. Now, there are considerations when assent can be waived under the U.S. regulations if the child's capacity is limited, or there may be a prospect of direct benefit that's only available in the research. In the recent FDA guidance, it was stated that children 7 years of age and older are often considered capable of assent. That is a true statement. This is often the age at which many research ethics committees would consider a child capable of assent. But this must be individualized to the age, maturity, and psychological state of the child who's enrolled in the research. Now, one of the challenges in the FDA regulations, as well as in the guidance, is that there is no statement of what is necessary for a child to be able to give assent. My own view is that we must interpret child assent in the context of parental permission. It's the parent who is making the judgment about appropriate risk and benefit, not the child. Now, child assent still remains important as well, but we're not expecting the child to be able to make an informed evaluation of the risk and benefit, but rather, does the child agree to participate based on the information that has been provided? Back in 1978, the National Commission, in a different report that outlined the essential elements of assent, but none of these, unfortunately, found their way into the regulations. What they stated was that the essential elements of assent for individuals with limited capacity would be to know what's going to be done, to be able to choose to, in fact, experience these procedures, communicate this unambiguously, meaning be able to say yes, and be aware that they don't have to do this. And if we view child assent from this more limited perspective, I would argue that many children ages 5 or 6 could, in fact, meaningfully assent to certain procedures, such as a simple blood test. So this has been a rather quick overview of the ethical considerations in pediatric clinical research, and I look forward to further conversation about this in the discussion time that's hopefully available within this session. Thank you.